And by now, some of you should have gotten, we got a challenge of our system that we're using from Mac. And that's why most of you have not yet received your feedback. That's why most of you, because of some technicalities in the within the Google Classroom. So it was a little bit complicated. Marking sometimes when you try to send back, what you have marked, so it doesn't go. But very soon, slowly by slowly, each one of you will be getting your what you got. But today, the main aim is going to be on how this question was supposed to be approached and the kind of probable answers that you are supposed to put. Now, I'm going to, my main aim here is I'm going to teach you how to answer paper 2 type questions. Depending. So we are going to see how you should have gone about answering this question. And then tell you that the next time you are given such questions, hey, you are able to answer something. You're able to answer something. Let me see. Time check is 11:04. Okay, I think we can start. Number one, what I want to comment about is some of you, when you are given this question, you didn't attempt from what you had done. Actually, some of you just copied from the notes direct without even changing anything, whether it made sense or it didn't make sense. So that is a challenge, which means that in case you are given a paper 2 type question, like in a paper or in an examination situation, it means you cannot answer. The purpose of this assessment was not to see whether we gave you notes and you can copy. No. Because you could see, and I was surprised that some of you can't even at least edit something like the way it was. You see, it is, of course, there are some people who have that capacity, but even the words be, sometimes the arrangement can't be exactly the same. So some of you, what you did was to get the other PDF which I sent, and then you just copied direct in order. That's number one, which means some of you had not understood the concept. But most of you, the good thing is that most of you, when I tried to read through the work which you sent, most of you were on track. And you understood what the question was all about. Because it was about describing the mechanism of water uptake. And I think I told you that to describe means like you paint a picture of something in word. Okay. So here you are supposed to give us the whole journey taken by water, right from the soil up to when it leaves the plant via the stomata or the stomatopores in the leaves. So that was the challenge that we had to take on. And of course, this question had just 12 marks. Now, you should take note when I'm marking. The marking guide will have more points normally. And for example, I can mark a script. For example, the answers when it has like, let's say, 20 marking points. Okay. Like when I'm marking myself here, I may get 20 out of 28. The question was asking for 12 marks. So which means that that marking guide is able to accommodate, because it has been limited to 12 marks, but it could have more than 12 marks, such a question. So, but you find that there are some people who may give certain answers and they may leave out some, and then they also get 12 out of 12. And I think last week you just witnessed a release of of the UNEB question. Marks. And of course, very many people celebrated. And for us in the biology fraternity, we're happy. One is the number of A's increased from about 3 years ago when there were only 5 A's in the whole country to now that we are getting very many A's. There were very many A's. In fact, some schools got like up to like 17 A's, okay, which is good. But of course, we cannot compare it with some other subjects whereby there are some schools whereby, like for example, in mathematics, they got like 140 A's. You see that? But if you are to compare it with the biology A's, I think the school which got the most number of A's had about 17 A's in biology. But of course, the B's this time were many. I know the school which had there were 58 B's in the whole class. They had like 58 B's, and C's you can't even count. You can't count. And so it was a good performance this time around. And if even you, where you are, you can put in some good effort, you can, when next year, is it next year or this year? I think next year, when you are sitting your UNEB, you may sit around July or something like that. You may also be among those who are celebrating with A's. But the challenge with getting many A's and B's normally is that the competition is now a little bit high for the medical courses, which many people who do biology are opting for. That is Bachelor of Medicine and Surgery, as well as pharmacy. Normally you find that it now becomes, competition becomes very, very stiff. Very, very. So if you are a mediocre and you get C and you celebrate, you find that you may not compete with people who are having an A. Okay. Now, and it doesn't, by the way, it doesn't matter which school you are coming from. It depends on your personal efforts right now. Your personal effort will determine, not saying that for me I'm in this school, blah blah blah. So that doesn't matter. Your personal effort counts, because there are some people who are lazy and they are in what you may call very good schools, but you find someone getting an E or a D. Okay, I need someone who is in a school which they think that the school is not good. They have gotten a B or an A. So it doesn't matter. It is your personal effort. So we are going to go through this question as you are supposed to do it. Okay. So we can start. It was saying that describe the mechanism of water uptake from soil to the leaves by the plant. And the note NB, you see, sometimes you can make a marking guide, and some points are left out. So during marking, when such a point, which part of the marking guide is seen and it is correct, yes, a mark can be given to you. Okay, so you will see when after going through that, some of you will be asking questions, this and this. What about when I say this? They all count. They all count, and it depends. So we were told to describe the mechanism of water uptake from soil. This time it is from soil to the leaves. Okay, that means if you still remember during the lesson, I divided this process into about 3 themes. Okay, whereby theme 1 was about uptake of water from the soil. Okay, then theme 2 was about how water enters into the vascular tissue. In that case, it was the xylem. And then theme 3, how the water moves up the plant. Those were the 3. So when you're describing such a question, it has to be in those 3 phases. uptake from the soil, and then how water enters into the xylem, because it also has some dynamics there. And then once it is in the xylem, how does it move up the plant? Okay, so those are the major three themes that you are supposed to take note of. Okay, and we can begin with here. Of course, we are beginning from the soil, and you know that water is going to be in the soil, so you can call it soil water solution. So plants have root hairs that penetrate between the soil particles and absorb water from the spaces, because what is going to take up this water are the root hairs. And that's why at one point when we said that when you're if you still remember when I was teaching, when you transfer a plant from one part of the soil to transplant it somewhere, sometimes it can die in the process. And the reason as to why it dies is that most of the root hairs are dead, and so the plant can no longer absorb water. So you need that aspect, that what is the main way of how this plant is going to absorb water is via the root hairs and absorb water from the spaces. Okay, so you get that one mark. So water is drawn, but of course you can miss out. That doesn't matter. There are more and more marking points. So water is then drawn into the epidermal cells because the root hairs have them. So water is drawn into the epidermal cells. That is the first root of the root hairs by the process is osmosis. Okay, but why does it enter by osmosis? Then you tell us the dynamic, that there is presence of sugars and metabolites. And what do? They lower the water potential of the cell sap than the surrounding of the surrounding soil water. Okay, and then water water molecules then pass across the cell wall. You see, we are describing step by step. So water molecules then pass across the cell wall and protoplasts. Remember, protoplast means the living contents of the cell, which are normally enclosed by the cytoplasm. That's why if someone, instead of using this word protoplast, if someone uses cytoplasm, they are also correct. And then when someone says water molecules then pass across the cell wall and cytoplasm, let me just put here cytoplasm, because in the cytoplasm, that's where you find the living contents within the cell. So this person is also correct, and into the subvacuoles. The water then passes to the vascular tissue. You are now telling us that passes through the vascular tissue via the parenchyma cells. You see, from the epidermal, have epidermal cells, then cortex, and then into the vascular tissue. You see that we are still the track, we are still describing, we are painting a picture of this pathway in words. And then because before it reaches the vascular tissue, there are three pathways. So there are three pathways by which water moves through the parenchyma tissue. So there are three pathways by which water moves through the parenchyma tissue, and these include and have the apoplast pathway, the simplest pathway, as well as the vacuolar pathway. You see, so already, even if someone just did this, they're having 1, 2, 3, 4, 5, 6.5 marks. You see, that's why nowadays biology was simplified, and it's not, unless you have not been reading. The challenge normally comes with people who don't want to read textbooks. They only want to read those pamphlets that have already made answers. Okay, but once you are reading a pamphlet with already made answers, your biology normally is shallow. your biology is limited to that answer because you've not read from the original text of the textbook. Okay. So normally for an A material, actually for any process that you are reading, it is actually always advisable that you read three texts per 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 per, per, per process. Like you can read from BS and you see what BS says about it. You can read from, let's say, functional approach. You can read from advanced biology by Tool or advanced biology by Simpkins. So 3 versions per text. That's what you need. And by the time you finish, even if you've not crammed those answers, you can be able to write this. Okay. So I think you can see how we have moved. Here, even if you get 4 or 3, still you can still get more, because I've already told you that we give an allowance of many marking points, and that is why it is always good that you write for paper 2. You are given booklets, right? Because the markers will have to read every word that you've written. And you never know, you may have put there some marking points. So always write, don't write small things. That is what normally makes people get small marks, because you can never know where the mark is. You write, as long as you know that it is biology, just keep on writing. Okay. So we had ended when we are saying that there are 3 pathways that is taken by this water to reach the vascular tissue. Emily is saying, so what if I apply the hypotonic and hypotonic, can't I get them? This is very good. You'll, Emily, you'll get a mark. Because hypotonic, we know what hypotonic means. For example, here, here where I said, but which lowers the water potential of the cell sap. So here you can apply those words of hyper and then hypos, they mean the same. Lowering the water potential. If you don't want to use this word lowering water potential, you can say which makes the cell sap a hypertonic relative to the surrounding soil water solution. You see, so you can use that word, which makes hypertonic. So I've already told you during marking all those ones, because for me I didn't want to over make the thing bulky. So here normally they will stroke hypertonic. Hypertonic. Okay. So you get a mark. You get a mark. It's very correct. Now, when water reaches, actually, I think at one time I will display for you the report which came from UNEP for paper 1, and you'll see. Let me see, teacher. When should we expect our results? Emerald, how do we get our results? You'll get them via, again, Google Classroom. After marking, we send back a copy to you via, it's still in the Google Classroom, and you're able to see what you have scored. So you'll get them very soon. And then when water reaches the endodermis, it cannot enter the vascular cylinder or the xylem vessel. So here you may not use vascular cylinder, and then you say it reaches it, or you can say it does not go via the apoplast pathway. Sorry, water moving via the apoplast pathway does not enter the xylem tissue. So that is also half a mark there. Okay, so it enters instead via the cytoplasm in the simplest pathway. Okay, resulting into high pressure, which push water into the xylem. Now here you are describing the root pressure You see, because the root pressure also contributes in this process of water uptake. So which push water into the xylem vessels and up the stem. Okay. Then endodermis also actively secretes salts into the xylem vessel, creating relatively low water potential. Now here, you can apply the other words of hypotonic. Okay. Sometimes some people use osmotic pressure, those things. But maybe what I can comment about that is very many students do not understand the usage of that word, let's say hypertonic and hypotonic. They confuse it. Okay. So if you know that you are going to confuse that word, please don't use it. Instead, use or even this word of water potential. Some students confuse. They don't know how to use the word. When do I use water potential? Water potential. When do I use solute potential? When they don't know. So if you are among those people who can't know when do we use the word water potential, or how can you that lowering water potential means this, or increasing water potential means this. If you're not sure of that, then use the word more concentrated or less concentrated, okay? Because that is a very clear word. So here you can, instead of using this creates relatively low water potential, uh, uh, low water potential in the xylem, and you can here use a word. This makes maybe the solution there to be more concentrated. You see, so you're using the word more concentrated instead of using those words of. And while say, teacher, sorry, but do you please help a bit to go through the simplest upper plus pathway, please? Emily. Okay. I don't know whether you guys have forgotten simplest upper plus pathway. Okay. Let me comment about it. Let me begin by drawing just a simple cell. Okay. Let me draw this. What I'm drawing here is a root hair cell. That is cell wall. Okay. And then what I'm drawing here is it's C cell membrane. Okay. You can also have more of other cells, and then you can have the cell membrane there. Let me also draw another one there, because some people are maybe a bit confused about what I'm talking about. So what I'm trying to draw is like a plan of different cells and arrangement. Of course, normally they should be a little bit many. Let me draw like let me draw about two to represent cortex. I'm assuming this is the cortical cells, and then from the cortex cells, then we shall go to the endodermal cells. These are the endodermal cells. These are the endodermal cells, and then you could also have thin cell of the pericycle, and then here that's when you can now, let me draw it like this. Let me draw it like this. I'm assuming this is the xylem. I'm assuming that is the xylem. I'm assuming that's the xylem. So let me also draw. What I'm drawing inside is now the cell membrane. That is the cell membrane. That is the cell membrane. Of course, that's the cell membrane. And then that's the cell membrane. And then that's a cell membrane. So so when you're discussing this, you must have this picture of a transverse section. I'm just doing a small bit of a transverse section of a dicot plant, a transverse section of the dicot plant. So yeah, okay, like that, like that, like that, like that. So so what do I mean by because you said what? Simplest, then apoplast. Now, when water, I'm assuming these are, okay, let me get, soil is normally black. Let me put these particles to represent this soil particles. These are soil particles are there. So particles are there. And then let me get the blue to represent the water. So you're going to be having water is there, water in the soil. So this is the soil water solution. Of course, the soil water solution may have some minerals in it and other things. So so you need this water to go from the soil water solution to reach the xylem, if I can call this xylem. This is the xylem. This is going to be the this is the epidermal cell layer of epidermis. And then from the epidermis you have cortex cells. These are the cortex. Cortex cells is majorly having just parenchyma cells. Parenchyma cells just pack up, they fill up space, and they can even store food and all those things. And then this is the endodermal. This, you must have this arrangement. This is the endodermis. Endodermis. So from the endodermis, that's when you go to this small layer of the pericycle, and then to the xylem. So this is the route that water must trek to reach the xylem tissue. Okay. So what happens is that normally you're going to have water taken through. So the first pathway, and the one that takes 90% of the water, is you see, I gave an example last time when I was teaching this, that the cell wall is like those Crocs. Crocs are those shoes which are now on market, and I'm seeing very many students putting them on. Those Crocs have those holes in them. Okay, they have holes in them. Holes means obotuli. They have holes in them. So the cell wall is freely permeable, like water can just enter. If you are putting on a Croc, pericycle is here. Pericycle. So when you're putting on this Croc and you step in water, water will just enter, and all its particles, like things can just enter. Okay. So things can just enter. So what happens is that this cell wall is freely permeable, and so 90% of the water is going to take via this butuli in the cell wall. So water just enter through the cell wall like that until those spaces, into the spaces of the cell wall, like that, like that, like that, like that, like that, until it will, until it will reach here. Let me first erase that. Let me first put a Casparian strip in this endodermis. That's a Casparian strip. This thing is moving all around the whole cell, just like a belt. It is around the whole cell, just like a belt, and water that is moving through the cell the spaces in the cell walls cannot just continue when it reaches the endodermis. No, it stops here. It stops here. It doesn't cross. Okay. So the spaces within the cell wall, between the cell wall. So those spaces, okay, that is the apoplast pathway. Now the other pathway is what they call the simplest. Simplest, if you always forget it, remember simplest like cytoplasm. That is the route that goes via the cytoplasm or the living contents of the cell. That is the protoplast. So water will enter like that. Hey, let me get in a different color, probably blue one. Let me get a blue one. Yes. So water will cross like that, just through the cytoplasm, and then like that cytoplasm, like that, and then it will reach this one is not blocked, but of course it goes via this membrane of this, which is selectively permeable, the cell membrane. So that's why less water goes via that pathway. But water which is going via the apoplast pathway, once it is blocked by the Casparian strip here, it instead diverges and then begins taking the simplest pathway. Okay. But then there's also another pathway, which is the vacuolar. Now, in this case, I can draw, here I can draw another vacuole, the tonoplast that surrounds the vacuole. Okay. There I'll have to draw in another the tonoplast here. Again, I can draw another tonoplast here. Now that is the vacuolar pathway. So it will go through it is the same as actually the simplest, but the only difference is that here water can also go through the vacuole and then like that, and then like that. If there is another tonoplast there, and then it will also go like that, if there is another there, because each cell normally these plant cells have it like that until it reach there. So that's it. So the xylem vessels are narrow. And a high adhesive force develops between the water molecules and the lignified walls. Okay. Raymond says, what if you draw a diagram also, sir? Here, if you draw it, and you put a key, if you draw a diagram and you put a key on it, for example, if you do such a diagram and then you label it very well, and you put a key that maybe this arrow, and in this case, because you are using the same pencil, so you may put maybe stars on this, you put a key to show the different pathways, then that's it. You may get, if you have not written any word and they find this there, they may give you a few marks. But of course, the diagram does not explain it all. Okay. So paper 2 will majorly, like, unless they have told you with illustration, like the other question which came, there's a question which came some time back when they told candidates with illustration. Okay, their diagrams will carry marks. But in most cases, we want you to write words. We want you to write words. So, but if you put a diagram, it may not carry more marks. Most of the marks are going to be placed on the descriptions. But in case you had drawn a diagram and you had labeled it they may put some marks on it. Yeah. But most are going to be on description. So I encourage you to always write and write and write a lot, because that's paper 2. That's paper 2. That's its setting. So water, the xylem vessels. Okay, the xylem vessels are narrow, and a high adhesive force develops between the water molecules and the lignified the xylem. Okay, let me first get a red pen here. So telling us that they are narrow. Of course, here the water molecules develops, a high adhesive force develops between the water molecules and the lignified. Okay, here was because it has an effect. So effective evaporation of water from the leaf surface causes transpirational pool. So evaporation causes the transpiration pool, which pulls water. The water moves through the xylem vessels. So here you're again describing it, that it is moving through, excuse me, it's moving through the xylem vessels and tracheids. Remember, the xylem is made up major water conducting tissues. That is the xylem vessel, or sometimes called the vessel element. So in case here, instead of writing xylem vessel, and you write vessel element, vessel element. Okay. I'm skipping the word element. Vessel element. Okay. So when you write vessel element, and all, can either use and all tracheid. Because the water conducting tissues. Okay. Or even if you just write through the xylem tissue. Okay, that is okay. But to be specific, you write the vessel element and the tracheid. But sometimes the vessel element can also be called the xylem tracheid, and also this tracheid can be called xylem tracheid and then xylem vessel, or you can just call them by their names, vessel element and tracheid. This lowers the water potential. I told that if you cannot use this word lowering, if you don't know how to use the word lowering water potential, if you don't know, just use the word makes more concentrated. Because when you lower the water potential, you have made that solution to be more concentrated. So you can use that word. This lowers the water potential. You can say this makes the mesophyll cells to be more concentrated. You can use that more concentrated. If you don't know how to use that word of lowering water potential, concentrated, but just know from today that when you lower water potential, it means that that solution has a higher concentration of the solute. But if you increase the water potential, remember, water potential is a tendency or ability of water to move out of the system. And the water which has capability or ability to move out of the system, it means that it is there in plenty. It is there in plenty. So the solution with the high water potential, it means that it is having less solute. It means it is very dilute. And the solution which has a low water potential, it means that it is more concentrated with the solutes in it. And in this case, the solutes are normally sugars, which are made by photosynthesis. Okay. So this lowers the water potential at the mesophyll than in the surrounding cells, and more water is drawn into at the mesophyll cells by osmosis. You see, already someone was given that account. You already been having your marks. So the mesophyll cells are perforated by numerous channels which open at the surface. If you don't want to use this word, you can say that yes, they're perforated by numerous channels which open at the surface as minute pores, or you can call them stomata. Okay. It is perforated, which called stomata. Considerable adhesive forces develop um, develop in the narrow channels, and water is pulled up and prevented from falling back. So here you are showing us that adhesive forces are playing a role in preventing water from falling back. Also, the high cohesive forces between the water molecules which flow in the xylem vessels and patches of leaves. So here, high cohesive forces which flow in the xylem tissue from breaking. So you're telling us these cohesive forces, which are in between the water molecules, they are preventing this column of water from breaking up as it is moving up. You see that. Teacher, how does the transparency prevent out leakage of water that has already entered the xylem? I'm not getting it because the Casparian strip, you can think of it as just like you imagine. I'm assuming there are three holes through which water is entering, and then in one of the holes you put there plaster, which does not allow water to go through. Okay, so that the water that would have been taken by the three holes is now channeled to go through via one hole. You see that? So that is the Casparian strip. Casparian strip is going to it is going to it doesn't allow water, it blocks that pathway. But Arnold, when you ask that, how does the Casparian strip prevent out leakage of water that has already entered the xylem? Now, in this case, it's like you are saying that if you are having a plant, then sometimes, okay, if let me first go back to this picture which I had drawn. So because water that has entered the xylem here, it is rare. It is rare that water is going to come back from the xylem to go back this side of the cortical cells. Okay, and you know the reason why. Already, this plant is in the sun, experiencing transpiration, so water is being taken up the plant. Okay, so how then is water going to move? Maybe when you are creating artificial situations whereby you're going to place these roots, but of course this plant would have already died. Maybe you're going to place these roots in a high concentration saline solution. For example, in the case of people who do hydroponics, there in UK they discovered a situation whereby you don't need to grow plants in soil. Okay, so they get water and they place their mineral ions, and these plants grow in just in water, but that water normally contains ions in it. That phenomenon, I think, is called hydroponics, growing plants in water in a solution of nutrients. Unless you are looking at such a scenario, which is normally artificial. But for a plant, it's not going to be easy for water to leak from this side of the xylem to go back to the cortical cells. Okay, because most of the time, due to transpirational pull, the cells this side are having a low water potential compared to this, and even this. Going back to your question, this endodermis, where the Casparian strip is, has an element of actively secreting ions into the xylem, thus lowering the water potential here. So if the endodermis have that capacity of secreting actively, that is using energy, ions into the xylem vessel, making this the solution here to have a low water potential. How then is water going to move from this side into the cortex? Okay, just know that the Casparian strip is impermeable to water. It is impermeable. So because it is impermeable, it will not allow water to just move from the cortex via this apoplast pathway. Alternatively, if you want to talk about from the xylem, then it will not allow water to move from this xylem via the apoplast pathway. Okay, and then so this breaking up this also aids up water transport of water in the xylem vessels of the stem. And yeah, so this is kind of a pictorial view of this thing, okay, whereby you are having the root hair penetrating the soil particles, and there's a film of water there. And then you can see that they have three layers: the cell wall, the cell membrane, and the tonoplast. You see, so all these plants normally have it, and that's the arrangement of epidermis, the root cortex, and then the endodermis, where we find the Casparian strip. Normally, where we find the Casparian strip. And then pericycle, normally the pericycle, its function is to give rise to new root hairs. Actually, the pericycle produces the new root hairs. That's why it doesn't have a lot of function in water uptake. That's why here they ignored it. Emerald says, please pardon the last part of the previous slide. Okay, I'll talk about it. So that's why here they didn't include the pericycle, because its major work is to give rise to lateral, those root hairs. Okay, and then you can see the endodermis is connected to the xylem vessel, which goes into the leaves, penetrates by the midrib and the veins. And then those veins are normally surrounded by the mesophyll cells, which include, if you still remember from your O level, we have the epidermis, then the palisade mesophyll layer, the spongy mesophyll layer, and the lower epidermal cells layer, sorry. So where you have the stomata. And so once water has been lost by transpiration, the water potential here in the surrounding mesophyll cells is lowered. So water is going to move from the xylem within the veins and midrib to enter these cells. But remember, this water in these mesophyll cells is continuously being lost by transpiration. So that is going to create tension between the xylem and the mesophyll cells. Tension is a force which tends to pull things apart. So as tension is created between the water molecules here in the xylem as well as in the mesophyll cells, so water continuously moves up. And also that is the transpirational stream or transpirational pool. And also this endodermis, because it can actively secrete ions into the xylem vessel, making this solution here to have a low water potential. So it creates what we call root pressure, which continuously diverts water that is moving via the apoplast pathway, simplus and vacuolar, to go very fast into the xylem, and then water moves up very fast. That is the root pressure created by the endodermis. Jaden P says it's about the use of the Kasparis banana. You guys, is your network a problem? I've repeated myself for this. Okay. So has it is not the one which the endodermis, the Casparian strip is impermeable. Okay, it's just like seeing a leakage and then you put there plaster. Okay, so the Casparian strip is impermeable to water, and it is found only in this endodermal cells, because these endodermal cells normally they go around the transverse section of the what of the dicot in this case. So the Casparian strip does not allow water that is traveling to enter the xylem vessel via the apoplast pathway. And 90% of the water is actually taking this apoplast pathway, since the cell walls are freely permeable to water. So they don't meet any resistance. So once it reaches here, so all the water that is moving via the apoplast pathway is diverted and instead goes via the simplest pathway, that is through the protoplast. Okay. This last part was about cohesive forces. That cohesive forces are those forces between the water molecules themselves, and they just simply prevent the long column of water from breaking up. from breaking up in the middle, because that can be dangerous. It can kind of to transpiration, yes. So this also aids up. This also aid up water transport of water in the xylem vessels of the stem. And remember, the main force that drives water to move up the plant, the stem, is actually the transpirational pull. The transpirational pull, which I've just explained. So unless you have any other question, I've just gone through this. I've just gone through. Unless you have any question concerning this, but I've just gone through, and you should have been able to get all those 12 marks out of this question of describing the mechanism of water uptake from the soil to the leaves. If someone had understood, by the way, this last year's paper 2, it had number 1 was came from this topic, by the way, paper 2. If you read through paper 2, last year's paper 2, all look for last year's Waukesha mock. Last year's Waukesha mock, and do it. Try to do it. Last year's Waukesha mock. It has elements which are similar to this Waukesha, as well as the UNEB that came last year. So maybe give us a chance to draw the summary, that last drawing. What do you mean? This last one, this one. But it's almost time. We are left with this 6 minutes. So in this 6 minutes, and by the way, this picture is there in some of the textbooks. You open Advanced Biology by Simpkins, it is there. Even FA. The problem is some of you fear opening textbooks. You fear opening textbooks. These things are there. These things are there. Open those textbooks. They have some these things. Don't fear opening textbooks. Yes. Just screenshot. Okay. Unless there is any question for us, we are going to end the lesson. Is there any other question? Teacher, slide, please show. Slide 3. We are concluding, my friend. We are concluding. Is there any other question? Is there any other question? Okay, Admin Bernard, I think there are no questions. You can end the meeting.